Hello, folks. It's our last meeting of the semester, our last video of the semester. So you've made it through 15 weeks after you've watched this and completed uh, your reading for this week. It'll be the last reading, and you will only have your final exam to complete. And that final exam will open up this week, at the end of this week, and you will have until I think like next Tuesday or Wednesday to complete it. All right. So the lesson for this week is on bioarchaeology and forensic anthropology. And they're very these two subdisciplines are related to one another. Specifically, sure I'm not in the way of anything. Specifically in their kind of focus on skeletal analysis. So bioarchaeology is interested in human remains in an archaeological context. And so in this case, human remains are being used to understand how people in the past lived their lives, to get insights into culture, into social practices, and into the individual stories of those who those remains belong to. For forensic anthropology is focused on human remains, human skeletal remains in a legal context. So one of the big differences is in like how old are the remains, but both focus on looking at the bones to tell a story, to understand who this person's life, how they live. But the questions that we ask of the remains are different. So when you are doing either bioarchaeology or forensic anthropology, my head isn't the way. Let me move myself over to the other side. When you're doing bioarchaeology or forensic anthropology, there's a series of questions that you're asking. So the first is what I'm looking at actually bone, or is it something else? If it's not bone, then you're not going to ask osteological questions of it, right? So you're not going to answer bioarchaeological questions or forensic anthropological questions. The next question you want to ask is it human? For forensic anthropology, it needs to be human in order to do forensic anthropology, because you're trying to understand, you're trying to basically figure out who this person is, why they died, and whatnot. For bioarchaeology, you want to know if it's human for two reasons. One, if it's human, then you can ask questions specifically to that. If it's not human, how is it related to humans? So it might be animal bone, and so say it is the femur of a deer. So that's your long leg bone in your upper leg. So of a deer, right? If that's the case, if it is, why is this associated with human activity, right? Was it hunted and butchered? You know, what's going on? Those are still interesting bioarchaeological questions. They're just a different kind of question. Is it modern or archaeological? So is this something from the far past, or is it more recent? If it's modern, and by modern we mean like in about the last 50 years or so, then there may be some kind of criminal context associated with it. And so a forensic anthropologist would want to analyze those remains to figure out the criminal context through which with which those remains are associated. If it's archaeological, then a bioarchaeologist is going to want to look at those potentially and consider, what that might tell us about how people lived in the past. When you're doing archaeological research, the first thing you need to determine is if you have an assemblage of bones, is how many individuals are present. And so you'll want to calculate the minimum number of individuals, or the MNI number. So imagine you find an assemblage of bones and you see one skull and you see a set of ribs, but you see two right leg femurs. So two bones from the upper right leg. Well, you know that you only have one upper right leg bone, right? And so there's at least two individuals here, even if the only kind of duplicate is that one femur. Especially for forensic anthropology, you want to figure out who this is. Forensic anthropologists work on skeletal remains, and so this often means you don't have very much soft tissue, of any soft tissue left, and so you're trying to work on just skeletal remains. This can be really challenging. And so you want to figure out who this is. And so we're going to look at some examples later on about what you do to kind of figure out. And of course, you can use databases like missing persons databases in order to kind of further help. But your kind of primary goal as a forensic anthropologist is to figure out who you're looking at. Then from there, you look at pathologies and cause of death and whatnot. Is there evidence of trauma before, antemortem, around the time of death, perimortem, or postmortem, so after death? And then what happened to the remains after death? That's postmortem changes. We move knee over here with these bones. So in the initial skeletal analysis, is it bone? So bones can sometimes be difficult to identify, especially when you have fragments like you can see around my head right here. And things like shell resemble bone. There's some ceramics that can look like bone. And so your first goal is to figure, am I looking at bone? And really the kind of easiest way to do that is to look for the kind of two distinct layers. So you have cortical bone. Cortical bone is like the outside of a bone. So if you were to eat chicken, right, a chicken leg, and you look at the chicken bone, that outside of the bone is called the cortical bone. The trabecular bone is the spongy bone that's in the middle. So if you were to break a chicken leg in half and look in where like the bone marrow and stuff is, that's the trabecular bone. So if you've got those two features, you know, okay, what I'm looking at is bone. Now you have to figure out if it's human, right? If you find remains and it's deer or it's a dog or whatever, this is not of interest in a legal circumstance. And so you want to figure out if the remains you're looking at is human. So bone size and shape are important. Forensic anthropologists are well skilled at analyzing bones and telling them apart. Of course, we can run genetics on stuff now if we need to, but most of the time that's totally not necessary. I'm not a forensic anthropologist, but forensic anthropologists are trained specifically in the identification of human bones. For here you can see this is a cross section of a bone here. For human bones, the cortical bone is a lot thinner and the trabecular bone is a lot kind of it's more pronounced in the in human bones. And so that's one way to do it. Of course, different especially if you need fragments, those are ways, that's one way to do it. But if you've got whole bones, right, different animals have different bone shapes. Move me back out of the way. You're going to look at, to answer the question, if it's modern or archaeological, obviously we can do soil analysis. When we looked back at the chapter on fossils, just think about that and all the factors that go into affecting, bone decomposition and stuff. And we can do soil analysis. We can look at the objects nearby. But if you find modern dental fillings, enamel dental fillings, that might be indicative of a modern skeleton because the new enamel, like tooth colored fillings, are recent. If you see silver fillings in teeth, then you're probably looking, it could, we still do silver fillings, some people do, but that could represent something that's a bit older also, so you got to figure that out. Dental wear, you can look at material in the teeth, the stuff that people eat, lots of stuff can tell you if it is modern archaeological, when it's the kind of stuff it's buried with, if there's any clothing associated with it. If you're at a site and that site is a known site from 2,000 years ago, and this is a sufficiently decomposed skeleton, then you can say, okay, this may be associated with it. You want to do some more work to verify that. Move me back over to this side. So you always want to figure out your minimum number of individuals, even if you're pretty sure you've got just one individual. And the way you do this is you take skeletal remains and you lay them out. You go, okay, I have two humeruses. I have two ulnas, one left, one right. Humerus is one left, one right. I have one cranium. I have one mandible. That mandible is associated with that cranium. And you lay the skeletons out on a table, and then you basically start counting bones. And when you have commingled burials, like you see up here, this assemblage of this is a mass grave. it can be a lot more difficult to determine minimum number of individuals. So it takes a really careful excavation. As you can see here, they've got it gridded out, and they have each individual numbered here. And then it's something like a mass grave where people are murdered and put into a grave. In that case, many times you're just counting skulls because a person is killed intact and then they're put in the ground. So you can count skulls, and this can give you an accurate account. But in other cases, so say you're at a bioarchaeological site, it is a predator den, say it's a hyena den, and you're pulling out, bones, and you identify all the human bones, and they're dating to, 40,000 years ago or something. And in that case, it can be a bit more challenging. You can't just count skulls because hyenas don't necessarily always drag off the whole organism, and so they may have like torn off a leg or whatever, and so you have to do the work to figure out the minimum number of individuals. Then you want to, once we kind of figure out how many people are here, which are the human bones, which are not the human bones, then you want to construct a biological profile. So if you're doing, especially if you're doing forensic anthropology, is especially important because you are trying to identify who this person is so that you can notify next of kin, figure out, knowing who they are can tell you a lot about why they died. And of course, just closing missing persons case and giving families closure. So we'll talk about each of these in turn. So I won't talk about them here. So when you are really kind of one of the first things you do, and this can go, there's two things that often forensic anthropologists start with, either determining the sex of the individual. I'm like in a disco. This is cool. Either determine the sex of the individual first or the age of the individual first. Both of those are, determine the age is more straightforward. Determine the sex can be a bit challenging, but you'll look at the pelvis and the skull. There are key features on there that can give you a clue as to whether it's male or female. Things like the mastoid processes. If you remember back to your hominin evolution lab, when you looked at all those features, some of those are indicative of male or female. You can look at, we can look at kind of sexual dimorphism in these traits, like the size of the mastoid process, brow ridges, the shape of things like of the pelvic arch and malformations, not malformations, but changes into the symphysis on the pelvis. Metric and non-metric trait, these are just things that are measurable. And then looking at sex and gender and understanding the social context in which this skeleton is found. So if you're doing bioarchaeology and you know that both men and women are engaging in heavy labor in fields, in horticultural fields, you may have less sexual dimorphism in some traits. If you have a society where there's a sharp distinction between what men and women do, then you may have more sexual dimorphism in the skeletal remains. And you understanding that social context plays a huge role in being able to identify whether the individual is male or female or man or woman, depending on the kinds of questions that you're asking. And of course, for forensic anthropology, in order to determine who the person is, you'll want to figure out, whether they're male or female, because you're going to look through databases in some cases to figure out who's missing. Just by determining if it's male or female, you're cutting off a fair portion of the possible number of individuals this could be, so that you have a narrow range to look through. It just makes identifying the person much more go by much quicker. So you can look at things like the skull. So cranial features on the left-hand side on the picture, you can see a male skull. And not all male skulls look the same, by the way, and not all female skulls look the same. But when you're looking at them, oftentimes what we'll look at is I don't know if you can see, here we go, I'll put it right here. So you've got your mental eminence and your mandible here. So the robustness of that can be really indicative of whether it's male or female, although there's a ton of overlap. You can look at the size of the brow ridges here. So these are the brow ridges. whether this is angular, so if you rub inside the the eye orbit, if it's sharp, that tends to be a feature that we see more in female skulls than male skulls. And then one of the key features that we'll use is the mastoid process. So the mastoid process is this right here. So there's the ear hole right there, the meatus, and then this right here is the mastoid process. So as you can see in the skull below, this is, you can see a much more pronounced mastoid process right here on the skull on your screen than you can here, right? This is probably a female skull. We've got a really sharp margin right here, small brow ridges, a small mandible, and very small mastoid processes. So this is probably a female skull. It's more similar to the skull you see on the right, although it's certainly not identical. The pelvis is probably the most helpful at identifying sex. There are, you can see the subpubic concavity. There are the ventral arches present, which I have arrows on all of these. The width of the pelvic arch. I don't have a pelvis, otherwise I'd show you. So where like the pubis is, the pubic bone, kind of in the middle and the front. If it's narrow, it's more likely to be a male, and if it's wider like this, it's more likely to be a female. It's especially easy once you have, if a female has had a child, because there are changes that happen to the surface of the symphysis, which is due to the stretching. There's cartilage between the two sides of your pelvis in the front, and when you have babies, that gets stretched, and so it modifies that, where you can see actually here. So you see where it says the istiopubic ramus, where it's pointing down below, that kind of flat surface just above it, that is the symphysis, and that can be really important for identifying female, especially if they've had childbirth. Move me over into this. Determining ancestry or race or whatever is something that's incredibly difficult to do, and is oftentimes not done so much anymore, mainly because it slow you down more than it can help, because they're like, yet again, races aren't biological categories, if you remember back to a few weeks ago. And while, as from last week, while you can see that racial categories have biological consequences, there's still so much overlap between human populations, or whatever you want to call them, that it's not particularly, there's no like traits that are unique to like Africans or Europeans or Asians. And so it doesn't work really well. So sometimes if you've got some sufficient amount of cultural context, so say you're like in a region that has a large African American population, and there are some clear, within not being African, but within that group, that localized group, there are some shared similarities, then you can maybe use it. Otherwise, it's really difficult to determine ancestry. Mainly for lots of reasons. One, race isn't biological, but like the environment, the kind of labor you do, how you live your life, all changes your body, and that stuff shows up. And so it's easier to infer like the kind of job somebody had than it is to infer ancestry. But we still, in some cases, we still try to. These are some of the features, like the transverse palatine suture in your palate, the hard palate, and the roof of your mouth. There's some dental, like a shoveling of teeth, which are more common in Native American people, although certainly are not unique to Native American people. Measuring the distance between the eye orbits can be indicative, and yet again, these are like statistical probabilities. So it's not like this, a broader interorbital breadth is indicative of being Asian. However, it might be more common in that population. Age is much easier to determine. It might be the easiest trait to determine, mainly because we can look, as you can see here, in the skull. In the bottom here, you can see all the teeth. This child so has deciduous teeth. Those are teeth that fall out, just like deciduous trees or trees that lose their leaves. Here you can see a growth plate. So this is an epiphyseal plate, and you can see that there is a gap between it. This is the soft part, so the entire bone has not calcified. So kind of up above the red arrow, that is bone, calcified bone, and below that, that is basically cartilage that is calcifying as the bone continues to grow. This is what we call the growth plate. And different growth plates throughout the body calcify and fuse at different times. And then the skull sutures. So if you look here on this skull, you can see these lines. This is a coronal suture, the sagittal suture. So these sutures, they fuse together at different times in fairly predictable and regular rates during development. And so you can look, if you have a whole body, you can look at all the fusion rates, you can look at the teeth, you can look at dental wear, and then you can get a fairly accurate age of a skeleton. Yet again, you can look at the symphysis, which is what I talked about earlier, and it degrades over time, and that can give you an idea about how old somebody is. As you have degeneration, you can look at the sternal ends of ribs. This is the part that's like right here at your sternum, and you can see that over time, you get some degeneration that happens there. Stature, usually what you'll do is measure the length of long bones, so bones that are indicative of stature, to try to estimate kind of a range at which the height of this person will be. That's part of the reason that your height needs to be accurate on your driver's license, because if you go missing, it makes it easier to just identify you if something were to happen. And of course, we have individualizing characteristics. These are things that are like implants, that have serial numbers on them, which are directly tied to you. Anything that dental records are another individualizing characteristic. Anything that is like something special about your skeleton that is documented in medical records can be thought of as individualizing characteristic, and these are by far the most helpful way to identify without having to correlate between lots of different missing persons reports, and then of course the data from the skeletal analysis. So once you figure out who they are, so you've gone through all of that information, and now you know who this individual is. Now you need to figure out how they died. In some cases, you do trauma analysis. Most cases, you're doing trauma analysis to figure out how they died. But so my mom's dad was murdered. And part of the reason they were able to identify his skeletonized remains that were identified by a forensic anthropologist, move me over here, was by looking at some dental records, but also by looking at some remodeled broken ribs. So he had broken some ribs. Those were documented. When bone heals, it doesn't just go back to perfectly normal. There are telltale marks of remodeling that occurs. And so if you have like broken ribs documented, then you can use those medical records to then analyze the skeleton. So when we analyze trauma, we want to look at things that happen either ante-mortem. This is stuff that happens before an individual died. So like for me, I have like warped ribs on this side because I have really bad asthma as a kid. And because of that, my ribs are kind of mal-shaped on my left side. So that would be something that would be like ante-mortem trauma. Perimortem trauma is trauma that occurs around the time of the death, and this trauma could be associated with the death of the individual, and this is going to be especially interesting. Antemortem trauma is going to be interesting for identifying an individual, but perimortem trauma is going to be interesting for identifying how that individual died. And then postmortem trauma is going to be interesting for understanding kind of what happened to those remains once they died. So there are 4 types of trauma. There's sharp force trauma, which you can see here on the left-hand side. You can see that this person's skull was sliced through by something sharp, exposing the brain, but slicing away the bone. These can also be caused by like punctures. They can be caused by like there can be slashing marks. There's lots of different ways that sharp force trauma can kind of manifest itself. Right above my head, you can see blunt force trauma. This is caused by crushing force, and oftentimes it'll leave some like radial crushing kind of far out. So you can see a little bit of that here, kind of, oh, other side, that side. I'm mirrored on the screen. But anyways, on that side, you can see that there's some radial trauma. So these are both examples of blunt force trauma. Then you can have projectile trauma. This is caused by gunshots. It could be caused by bow and arrow, although it's less common nowadays. Or you can have thermal, which would be being exposed to high heat and stuff. So you can see above me up here that these bones are charred. They've been burned. So bone pathologies, these are diseases or illnesses or stressors that affect the skeleton. And so we can see these and kind of lots of different examples of it. So we'll look at a few of them. So we can look at things like osteosarcomas. These are bone cancers. So you can see a kind of really terrible example of bone cancers. Another disease that shows up in the skeleton is syphilis. So when syphilis reaches its tertiary stage, its third stage, it causes treponematosis, which is the resorption of bone in the frontal bone right here. So you get all this pitting in the bone. So you can have an individual that potentially died from syphilis, and you can see that with the treponematosis. Osteogenesis imperfecta, this is like brittle bone disease. My dad's mom's sister had osteogenesis imperfecta. So the bones are really brittle and they break, and individuals tend to be really small. Rickets, this is nutrient malnutrition, and it causes bowing of bones. So on the left-hand side, you can see no, that's got rickets too. It's curved. So all three of those on the left side are all bent. You've got those are leg bones. So the one on the left is a femur, and then your shin bone, and then your second lower leg bone. So your tibia and your fibula. And then in the middle, this is tuberculosis. It's a bacterial infection that often infects the lungs, but it can have effects on the skeleton. So tuberculosis is one of those diseases that really shows up in the skeleton. And then you have achondroplasia, which is a kind of dwarfism, which you can see above my head. So those are pathologies. Taphonomy is the process of what happens to remains after they die, or after it's decomposing. So once an individual is dead and is no longer developing, the next process takes over is called taphonomy. So these can tell you something about how long remains have been skeletonized, how long the individual's been dead, but I also tell you kind of what's happened to them, which can give you some context and identifying kind of where they've been. So here are just some examples. I'm going to move me over just a little bit. So on the far side, you have root etching. These are just roots that are growing around the remains. If you were to find these, even if they were at the surface and you see these root etchings, which you can know that these were remains that were at some point underground. On the top middle, you've got rodent gnawing. You can see it around the edge. On the right side where rodents are gnawing on the bones. This tells us that the bones were exposed at some point and that rodents had gotten to them. Weathering right next to me. This means that the bones were exposed to the elements, so it probably wasn't buried. Also, there's no staining from the soil. So these were bones that were bleached by the sun and left out. And so there's weathering that's occurred. We've already looked at burned bone above me. But you can obviously have bones were burned, and then carnivore damage. So this could either be due to being killed by a carnivore and then predated upon, or it can be due to being scavenged. So an individual is left out and some kind of carnivore got to them. And on here, you can see gnaw marks on the bone. All right, this is the final slide. In doing all of that, one of the most important considerations that we make is in the ethics of handling human remains and being respectful. So there's I could teach a whole class period and probably more on just the ethics of human remains. So I'm just going to be brief here because this is a bit of a longer lesson. So when you're working with human remains, you want to be respectful of the dead. These are real people who had families, who still may have families. So you want to handle them carefully. You want to be respectful. You don't want to contaminate them. You want to preserve them as best as possible. In regard to the remains of indigenous people, they are protected under a federal law called NAGPRA, the Native American Grave Protection and Repatriation Act. And this basically makes it a legal responsibility for anybody who disinters indigenous remains to then repatriate them, to give them back to an indigenous group. And so scientists can't just dig up Native American burials and just do science on them all willy-nilly now. There are agreements with indigenous groups. It's federal law that protects this. And so if you disinter indigenous remains, often what happens is they just get reburied. And then usually the appropriate thing to do would be to then write a report communicating their remains here. And then the site may be consecrated again by local groups who find that site relevant to them. In terms of human rights, forensic anthropologists will often work on genocides. Kathy Reichs, the author of the book For Bones, who I worked with at University of North Carolina at Charlotte. Was a key anthropologist in both the Guatemalan genocide, identifying individuals, and also 9/11, identifying individuals from 9/11. And so forensic anthropology and bioarchaeology have very stringent ethical standards. I encourage you to go out and, if you're interested in this stuff, read about them. They do a very comprehensive job outlining the ethical standards, as anthropology tends to do nowadays. But I don't want to take up like another hour going through them. But I do encourage you to check them out. You can just type in forensic anthropology ethics, and you will be able to read all about them. But anyways, that is our lesson and our final lesson for this semester. So if you have any questions or anything, please reach out to me. Your final exam, again, will be available to you on Thursday, and you will have until like Tuesday or Wednesday of next week to complete it. All right. Take care.